So the first speaker in our session, we have 3 long talks and 1 short talk. And our first speaker is Apratim Chatterji from ICER Pune. And he's going to tell us about underlying principles of chromosome organization. Welcome.
Thank you. So first of all, thank the organizers for giving me an opportunity to present my work here. So my formal title is Topologically Mediated Organization of Equally Chromosomes in Slow and Fast Growth Conditions. And I shall explain what this means. But I could have also just equally said that my title is Underlying Principles of Chromosome Organization. And particularly in this talk, I'm talking about bacterial chromosomes. So first of all, why is chromosome organization important? I mean, there might be some physics students in the audience. This is basically for them. For the biologists, I don't need to tell. But the point is that different cells have different chromosome organization. And one could equally say that depending upon which, because each cell needs a different set of proteins, different set of genes need to be expressed, and a gene expression is often, so whether a gene is active or not is often decided by this promoter and enhancer sites. It's some lock and key to activate the gene. And this in turn, the enhancer can be often quite far away from the gene, and this can create a loop. And this is what I mean by a topological modification. So if different genes are active in different cells, then you would have essentially a different set of loops or a different kind of loops. And that in turn could also, so you will have a different chromosome organization, and that in turn will decide the cell function. So gene regulation and chromosome organization is strongly interrelated. And I particularly look at topological modifications, which means loops. So so I'm basically a polymer physicist. I tune entropic interactions between polymer segments by introducing well-designed loops. And once I have an understanding for it, sometimes I use that to explain chromosome organization. So my talk will be giving biological problems, but then going into polymer physics and coming back and explaining that. And I shall explain what I mean. In particular, what I'm going to talk about is spatial reorganization of tagged loci of fish of E. coli chromosomes in slow growth, and explain that using very simple polymer physics based models. The fast growth work I'm not going to talk about, which I had planned initially, and there's a poster by my student Sriyang Pandey. Instead, I'm going to talk about the underlying mechanism of transport of ODC from old pole to new pole in the secret centers. I mean, it is known that it is done by parabious set of proteins, but the question is, how do the parabias do it in a timely manner? This is slightly different. And if I get time, I'll also talk about spatiotemporal replicosome positioning, means the protein, the replication protein machinery in secret centers. And I don't think I'll get time to talk about it for E. coli, but I have data, and people who are interested, they can contact me. Now I'm talking about entropy, about cellular systems. Cell is a highly non-equilibrium system, right? So why do I talk about entropy? So it's true that the cell has non-equilibrium active processes, but the processes of, suppose, replication, they're often very localized in space and time, which means if replication is happening, it could be happening in 2 places, like 30 nanometer cube. The cell is huge. The chromosome is huge. So except for those active points, I could talk about a polymer, DNA polymer, which is in equilibrium. In particular, I incorporate non-equilibrium effects. I mean, I'm basically do polymer physics, but I do incorporate non-equilibrium effects of DNA replication, loop formation, chain crossing, which is the effect of ropoisomerase, growth of cell lengths, but they're all happening in localized point of space and time. And hence, for the rest of the DNA polymer, I can invoke concepts like entropy. And I do large mass simulations for those in the theoretical physics community. And so let me now give concrete examples of this. So the broad question in 2006, or even now it might be, that when there is a cell division, the chromosome, the DNA, has to be replicated into two parts, into two copies, two daughter DNAs, and they have to go to two different parts of the cell. Now, for higher organisms like humans and eukaryotes, they have a particular protein machinery which is called mitotic spindle. And they catch the two copies of the two daughter DNAs and pull them apart by force. Bacteria doesn't have that. This is a picture of the E. coli bacteria. It's at approximately 1 micron in diameter, approximately 2.5 micron in length. So it's a cylindrical molecule. For me, it's a cylinder. But some of them you see are much longer. And that's because they're about to divide in the middle. So this aspect ratio is around 1:5. But when it's divided, it's 1:2.5. But for bacterial, like E. coli, they don't have a dedicated machinery to segregate the DNA. They don't have a mitotic spindle. So the question was, how do they segregate? Once a DNA, the mother DNA is replicated, how will they segregate to two halves along the long axis? And the answer, and a proposed answer was given by June, Mulder, Wright, etc. long back. So they had a series of papers from 2006 to 2010, and they proposed that entropic forces in ring polymers, so bacterial DNA, which themselves are ring polymers, they could be using entropic forces to segregate their DNA. So now that's quite a radical idea. And I shall explain what that means. What do I mean by entropic forces segregating it? But this was already 2006 to 10. Right now, if you read any review papers, including the ones I have read by Lloyd, they say, okay, entropic segregation is important, might not be the whole story. There might be MOOCBEF, for example. And we shall talk about it. And what these people said before going to the physics were that, let me talk about the physics first a bit. So when we say that entropic segregation, so typically physicists or most scientists would associate entropy with mixing. Like if you have two gas molecules separated by a partition, by a wall, you remove the wall, they would mix. And why would they mix? They will mix to increase their entropy, they increase the number of microstates. On the other hand, if you have two ring polymers and they are mixed already, they would segregate because of entropy. Now, in this talk, I won't tell about the physics in detail because explaining the full physics takes 15 minutes, whereas want to. So basically what you have here is two chains, one colored blue so that you can visually identify them and another colored red, but they only have excluded volume interaction, means that two chains can't overlap. They cannot sit on top of each other. And that's all that they have. So they are ring polymers. There's nothing living about them. It's as dead as they can be. And basically, if you allow them, and they're highly confined, so inside a cylinder, the aspect ratio has been taken as 1:5, reminiscent of dividing equally. And if you basically allow Brownian motion to take over, you see that they spontaneously segregate. And they spontaneously segregate because of entropy, which means if they are segregated, they can take more microstates, they have more conformations. Now, to explain this will take 10-15 minutes. I'm not going to do it. I'm just going to ask you to accept. If you have questions, we can meet after the talk. But so here confinement was very important. And what the Sukjun and Wright and others proposed, Bella Mulder proposed, that if anyway two ring polymers will segregate because of entropy in highly confined system, it's highly confined for those who know. So maybe the E. coli chromosome also uses this principle to segregate its chromosomes. Of course, there are some issues, like there are some MOOCBEF, etc. If you don't have MOOCBEF, they don't segregate, but we'll come to that. But this is what the status was. And then there were some other experiments. Okay, so this is a picture which you are seeing for the third or fourth time today. So when we do polymer simulations and map them to chromosomes, what I want to say, we are not really looking at DNA double helix. So here you see this picture and this scale. So this is 2.5 microns. This scale is much bigger than the DNA double helix. And when we model chromosomes or when we associate our polymer simulations and claim them to be chromosomes. I'm really looking at large blobs of DNA inside which there is a lot of protein. So the length scale I'm looking at is typically 100 nanometers when I talk about it. So even while I'm mapping like the polymer chains to DNA, so I'm not really talking about the DNA double helix. I'm really talking about much larger chunks of chromosomes. So this is the picture you should keep in mind when I talk about the rest of the talk. And the point is, so till now I was talking about DNA segregation, but there is also evidence not only by Hi-C but also fish data, which I'm going to show here by Wiggins, that the DNA is not a random coil polymer. It has some organization. And that organization changes as a function of time, as seen through fish experiments. And I shall introduce what that is. So these are experiments. This is a snapshot from Youngren Genes and Development, June. So these are essentially, you're looking at E. coli bacteria under the microscope, where some segments of the DNA has been marked. They have been fluorescently tagged with fluorescent molecules, and you can see them green and red and so on and so forth. And so what people do is look under the microscope and see how this green and red and whatever whatever colors, they move as a function of space and time. And thereby they map out the chromosome reorganization. In particular, what Carson Wiggins did, 2016, that they marked 7 different points on the chromosome by fluorescent markers and looked at them in the microscope to see how they change as a function of space and time, as replication and many other stuff is going on. So to this audience, I shall not tell what is OD and TER. OD is the point where replication starts. TER, difter is the point where replication ends, and replication proceeds along the 2 arms of the DNA. So what is seen that if you mark the point called OEC, and this is the point where replication has begun, here it means that there are already 2 ODCs here because replication has begun. But at least in this picture, in this experiment, they see that the 2 ODCs are basically localized near the center of the cell. This white region is the length of the cell, and the cell length is increasing, is doubling as a function of time. And what it shows that the two red spots, the 2 ODCs are localized near the center of the cell, but around 20-25 minutes later, one of the ODCs, because they've been replicated, goes to the so-called quarter position, the other ODC goes to the other so-called quarter position. And this red color is a measure of the standard deviation. So basically, in a very, so this is, I think, average over 10,000 cells or so. So right around 20-25 minutes, the 2 ODCs split and go to the 2 quarter positions from the mid position. Not only this particular, so this is now time, this is now time, this is the length of the cell normalized, and it has these different points which have been marked. This is time relative to the time of split. So not only the ODC goes to the 2 quarter position, but 2 other loci like L2, it also starts in the middle of the cell and goes to its so-called home position. And this is a bit inside compared to where the ODC localizes. Some other guy, R3, this, it was initially all unlocalized, but then at some point, it occupies the middle region of the cell, but it is relatively delocalized. And some other point, like TER, it is initially delocalized or was near one end of the cell, and at some point called the TER transition, the same time when it goes to the 2 quarter position, this comes to the middle of the cell. This was understood actually even before we came. And the question is how and why? If proteins do it, how does protein calculate its time and take that particular loci to a particular point in the cell? In a very, okay, there are fluctuations, but still it's relatively, as you see, in a predictive manner. How does these decisions taken inside the cell? How do proteins do it? This is the same, it's data again from the same experiment, Carson Wiggins. So ORI first splits and goes to the quarter position, and different loci as a function of time, they go to their home positions along the, and this is the long axis, this is a long axis, and this is a function of time. So my aim is to explain this. How do there such spatio-temporal organization happen? To do this, what I'm going to do is to take a detour, take a digression, and discuss something else, discuss some polymer physics, just like I talked about ring polymers, and then come back and explain this data. That's how I'm going to do it. So I'm going to take a digression. So I'm going to talk about topologically modified polymers. So previously, Sukjun, when I tried to reproduce the data of Sukjun, I was talking about ring polymers. Ring polymers segregate along the cylinder. Here I'm going to play a game, and you should soon know why I'm playing the game. So suppose there was a 200 monomer polymer, I'm going to have a linker. Could be done by a linker molecule, but I'm doing a spring, so that you have a so-called a rotated 8 architecture. Now this has 2 internal rings. I can play other silly games like have 2 small loops, add 2 springs, so that you have 2 small loops and 1 big loop. And this I call arc 1 to 1 big loop, 2 small loops. So I can play these games. The first game that I play is, instead of taking 2 ring polymers, I take 2 rotated 8 polymers inside a cylinder. And now these 2 small rings, which are connected, are themselves smaller rings, which entropically repel each other. Just like rings repelled each other, the 2 small rings also entropically repel each other. And if you have 2 such polymers, each of these loops will occupy different points along the long axis. If you plot the center of mass of each of these loops along the long axis, you will see that this loop occupies this point, this loop occupies this point on an average. I'm plotting this distribution of the position of center of mass along the long axis. This guy occupies here and this guy occupies here. And moreover, if you plot the position of the center of mass of each of those 4 loops as a function of time, yes, each of these 2 loops can flip. So the green, which could be the center of mass of this one, could come here, but then the other one, this loop, then goes there. So they can keep on flipping, but they are very well separated in space. All the 4 loops are well separated in space. Next, I'm going to play the same game with another polymer topological, which has 2 small loops and 1 big loop. Now I'm going to have 2 polymers, but I'm also going to have an extra crosslink between these 2 polymers. So each of these polymers are like this. Each of these polymers are like this, and they are basically trapped inside the cylinder. And we have 500 monomers, and the way it has been made, that if this is monomer number 1, which later I'm going to call ORIC, and if this is monomer number 250, which is like left arm and right arm, I'm going to connect the middle of the right arm and the middle of the left arm to the monomer number 1 to create this. And keeping 2 polymers inside the cylinder with this extra crosslink between the 2 monomers at monomer number 250, which later I'm going to call the difter. So those are biologically motivated. What you will see that the polymers will organize like this. And the reason they will organize like this is there's a big loop of one polymer that entropically repels the big loop of the other polymer. This in turn repels the small loops of this polymer. This in turn repels the small loop of these polymers. Because of this crosslink, they can't overlap. So these 2 polymers, on an average, statistically speaking, will be arranged like this along the long axis of the cylinder. Why do you care? Give me one more moment. Let's talk about this particular monomer, which was cross-linked and is between this set of 2 loops. This monomer is essentially sandwiched between these big loops and these 2 small loops. A consequence of which this particular monomer gets localized along the quarter position along the long axis. So a particular monomer is also getting localized with fluctuations because of the design of this polymer. The reason you care is if you assume that a polymer has this particular architecture and they're arranged like this, then all the points L1, L2, L3, they actually got localized in space. So basically you have the two big loops here, but if you have a point called R1, then it will be localized near the poles of the cylinder. Some other point R3 will be somewhere in the middle because they are part of this big loop and there will be two of them. They are basically localized in the middle of the cylinder. I already said monomer number 1, which I shall later associate with OREC, is localized near the 2-quarter position, and things which these guys are localized. This guy is localized near the middle of the cylinder. Now, all these names L1, L2, R3, R1 are exactly the ones and in the positions which was there in the experiments. So the point is, if you assume that the chromosome, that the E. coli chromosome has this architecture, this is the mother, then the ori will be in the middle. And if these are the two daughters, then you can essentially explain all of the experimental data of Carson Wiggins and much more. And all that I assumed is that the topology is not ring, but two small loops and one big loop. Of course, to match the Hi-C data of your 2020, one needs a bit of more small loops, but none of these properties are lost. I'm not going to talk about the Hi-C data in the talk, but if you assume this, and you know what is seen is when you have the newly born cell, you have only one chromosome, then the ori, if you associate the monomer number 1 with ori, that's in the middle of the cell. When it has completely replicated, then they are in the 2-quarter positions, and they would be in the 2-quarter positions because you assume that the two daughters have also basically adopted this topology, and then you can explain this and all the other 7 points. So this till now what I was talking was pure polymer physics. It was like pure two taking two polymers, but E. coli also does replication and so on and so forth, many other things, chain crossing. So here what I'm trying to show that from a certain point here, this is just a schematic. This is not my simulations. This is just schematic because polymers are straight. I'm trying to show that the white mother is trying to become two daughters, red and blue. I'm just showing how we introduce. So suppose this was the mother and there was some cross-links. This is just a movie to illustrate. And you see the white is becoming along the two arms, red and blue, which could be the two daughter cells. So this is what we are doing in terms to model replication. And then when the point is replicated, you will see that some more springs which will appear here and so on. So this is the thing which I am doing. And if I put in replications, so now what I'm trying to say that there is already a polymer with the appropriate ARC12 topology, and that is becoming red and blue, which is the two daughter chromosomes. And you see that as white is converted to red and blue, the two daughter chromosomes, they are spontaneously segregating as the replication is going on. Here, there are 500 monomers, but here what we have done is show the same movie where I made the red and blue pictures, white spheres to be very small, and all that I'm showing is the two green are the oris, and the pink is the ter. And at some point, you will see that the ter, there will be a ter transition in which the ter will come in the middle and get localized, and the two green spheres will occupy the 2-quarter positions. And analogous to that of this. So this takes actually 2-3 times to go through, but you can see the localization. Here I showed only the ter and ori, but you can see all of them, all the different loci, and you can see spontaneous segregation. Actually cheated a bit. I talked about ARC2, that I was working with ARC2 in 2022. Since then, I have also introduced extrusion. And thereby, I can even control the so-called time of split or cohesion time. So here I'm again just illustrating the, oh shit, time over. Okay. Okay, so basically, if I introduce other details like effect of Mukbef, which is extrusion, I can even control the cohesion time. I can explain many other things like fast growth with the same topology. I can resolve contradictions between the train track model and the replication factory model, where the repliosome remains centered near the center. And well, I already showed that I can control different types of loci split agreement with broad features of Heisey map. Can I take 2 minutes? 2 minutes is okay? 2-3 minutes. Because I want to talk a bit about the effect of the PAR ABS system in the secret centers. Okay. Before that, I need to give this introduction that if you have two polymers like this, you know, there's an asymmetry. You have two small loops and one big loop. Are all the configurations, and there's only pure repulsion, there's no attraction. So basically, the C3 configuration is less probable than the C4 configuration, where I can draw arrows, the big loop and small loop. However, if you have 10 small loops on one end and one big loop, and this will be relevant for secret centers, then basically the C4 configuration is 5 times more probable than the C3 configuration. C3 is this configuration. So the small loops want to always face the walls. The big loops want to overlap. Coming to secret centers, it is known that the chromosome organization is very different from that of E. coli, and that one of the orices is localized at one of the poles, and once it's replicated, one of the replicated orices goes to the other end of the cylinder, of the cell, sorry, inside the cell. And this is maintained by the PAR-ABS system, and PAR-A itself has a gradient along the long axis of the cylinder. And it was already, so this data is from Christian Jakob Wagner. One ODC remains near the center, the other one basically zooms to the other end of the pole, and this is done by the PAR-ABS system. Again, the question is, how does PAR-ABS system know that it is to take one of them to the other end of the pole? And what I realized, I mean, though it is known to be a solved problem and known problem, what I realized is what PAR-A does is sit randomly along the DNA. PAR-B are special sites near the oric, where PAR-B gets loaded near PAR-S sites near the oric, and all that PAR-A does, PAR-A is somewhere randomly loaded along the DNA contour. When they come in contact with PAR-B, they get attached for some time. That is essentially forming a loop like the ones I already showed, like a lot of small loops at one end. And so I model just this. I don't know what is PAR-A, what is PAR-B, or what I had. So here, so these orange molecules are like PAR-A, which are loaded on the mother chromosome. There's a density gradient. And here what is happening is the white, which is the mother chromosome, is getting replicated to this purple and the blue ones. And if you notice, and the red ones are the two orices, here the transition has already happened. But if I run the movie, you will see that basically one of the orices hovers around here and then suddenly zips to the other end. That is exactly what is seen in experiments. And I have not put in any proteins. I don't know the properties. All that I have put in is effective loops formed by PAR-A to the oric. So this as maybe just once more. So if you just look at, Anyway, I think I'm way over time. I can show this to you after the talk also. So this is the experimental data of Christian Jakob Wagner. And the same thing where I just made loops from part A and it gets attached to part B. That's what I read in the papers. Can reproduce all the results. This is experimental data. This is par AB, no par AB. In one case, it zips past. I can also explain the replisome positioning using very similar principles, where basically what was told is the initial part, the replisomes are basically near one end of the cell, but towards the end, the two replisomes occupy the middle of the cell, and this automatically comes out from the simulation. I don't have to do anything except the paré gets attached to the orices. And there's a replisome video, but I can show this in person if you are interested. And the most important part is the students who did this job, all these different kinds of modeling, and that's why I'm giving the talk here. So thanks to them. And I can also give the talk because I've been trained in the biology or taught a lot of biology by a lot of people. Else it was not possible for me. So with that, I'm sorry for taking more time, and I thank you for your attention.
Okay, we have time for a few questions. This is a small meeting and we're not under time pressure, so we might as well enjoy the discussion, right?
Nice talk. Of course, your simulations are driven by the experimental data where you're introducing 3 loops, right? For example.
Those loops were to get the
yeah. So, but my question is that if you see, for example, that E. coli chromosome picture, the loops are of size 10 to 100 kilobases. So many loops are there.
Correct.
Do you think, you can probably make smaller loops of such size?
So that is what is already, so initially when I started it, I had only 2 big loops and I could get the fish data. Then I introduced 2 more loops in the ter region to go closer to the HiC map. And right now I am introducing many extruding factors of smaller loops near the proximal region. They are supposed to mimic the role of MUGBEF. And what they do is have a lot more loops and increase the entropic repulsion. That is why David Sherratt sees that without MUGBEF, which are extruders, they don't see it. So I don't know where they are sitting. At the moment, I'm doing it in a design manner. You introduce more loops, you can control the cohesion time, you can increase the segregation rates, and so on and so forth.
So another factor, I think, of course, it's not just the loop, right? Intertwined.
It's much more complicated. So those, I think what you have in mind is hierarchical loops. So I've already have the first hierarchical loop. There was the 2 small loops and then you had the extrusion loops. Those are systematically, we will be looking at hierarchical loops, but we are introducing complications. The Conrad-Waldring saw left arm and right arm, which is also relates to your data. They were in 2 different halves of the cell. Some see it along the length of the cell. So how to get that? That I can also explain. I know the principles. So somebody has to check, go to the E. coli, go to the secret centers and show that this really happens. So that is what I mean. I'm looking at the mechanism saying if you get this, this can be explained. But is what I'm saying, is that true? Somebody has to check. But by the way, those 2 small loops, there are RNN operons, which basically co-localize the middle of the left arm and right arm to the orifice. Sorry.
Hi, great talk. So you used the Langevin equation, correct?
Yes.
Now I wanted to get your opinion on this. What do you think will happen if instead of DNA polymers, you had RNA polymers? So they're also present.
Utman, I'm a poor physicist. At the moment, I can handle replication, which is a very huge complication, chain crossing, topoisomerase by having. At the moment, I can't handle RNA.
Okay.
I can handle 4 of the active processes.
So, Apratim, you mentioned something during your talk about the dynamics being non-equilibrium, but I didn't quite understand what role it played in whatever you told us.
So if you have long chains and you don't allow chain crossing, half of the time they won't segregate. They can become concatenated. So topoisomerase is important. I'm introducing loops even as I'm replicating them. I'm adding monomers. That itself makes it non-equilibrium. If you looked at the length of the cylinder was increasing to keep the monomer density constant. So, but I'm doing these steps every 10,000 or 20,000 iterations, a Monte Carlo step. So in between it is undercoats equilibrium. So that's what I mean, that even if it is highly non-equilibrium, and I do non-equilibrium stuff in 4 ways: replication, increase in wall length, cylinder length, chain crossing, and something else. Introduction of loops, intermediate in time.
Could the next speaker come get ready for their talk while we finish questions? Okay, Vicky.
Yeah, thank you for this talk. Very nice. I have a very naive question. Okay, so these loops, can we interpret them like unit tethering of the chromosome?
It could be it could be RNA polymerase getting co-localized. The one big loops. I mean, okay, so there was a ring, and then I put a cross-link. For me, there were springs. But in reality, there will be most probably long-lived co-localization of RRN operons, which basically are RRN, H, and G. And so the other smaller loops that I've started creating, for me, I'm just pulling loops. But that would be due to most probably MUGBF, because I'm putting them only near the ori-proximal region.
Okay, just related to this, do you think how the anchoring to the membrane would affect in your model?
Attaching to the membrane, for this I haven't done because I don't know about lamin proteins, but my student will be talking about similar polymers in a sphere, not associated, motivated by chromosomes. And at a later stage, of course, they will be important, but this is what we have just started and we won't be showing any data. So there will be things with membranes and without membrane proteins, but that's typically in a sphere, which is motivated by the nucleus. But it won't have one-to-one overlap.
One last short
Sorry.
question, because I'm dying to write on this blackboard. You had 2 loops, the way you did it, 2 small loops and a big loop. But what if you made this loop smaller and these loops sort of more equal? What does that do for your model? If you change the loop size so that this large loop is actually about, say, 3 equivalent loops, for example.
So in that case, the organization will change depending upon the design of loops. You can change the segregation rates. You can change the amount of mixing, demixing. So actually, I am showing you only one of these perfect things which matches with E. coli. We actually did 12 in 22 and 2022. And since then, we have kept on playing. We have a lot more experience with playing with loops. To give me an organization, most probably I imagine.
Okay, thank you again. Thank you very much.